How does colchicine work? Let's zoom into a cell. Normally in mitosis, these structures called microtubules, specifically kinetochore microtubules, attach to chromosomes via the kinetochore proteins to align them in the middle during metaphase and pull the sister chromatids apart during anaphase. Colchicine works by blocking these microtubules from forming, thereby disrupting mitosis. How are microtubules formed? Microtubules are polymers of alpha-beta tubulin. In normal assembly, alpha-tubulin and beta-tubulin come together to form alpha-beta tubulin dimers. These dimers add to the growing end of the microtubule during assembly. But when colchicine is added, it binds to free dimers at a binding site between the alpha and beta tubulin, forming this new tubulin-colchicine complex. This binding changes the tubulin dimer conformation from straight to curved. When these tubulin-colchicine complexes bind to the growing end of microtubules during assembly, this effectively caps the microtubule, blocking further polymerization and destabilizing the polymer. This is because the curvature of the tubulin-colchicine complexes prevents necessary tight lateral interactions between tubulin dimers. At lower doses, colchicine arrests microtubule growth, while at higher doses, it promotes degradation of microtubules. Let's look at the effects this has on a cell. In normal, untreated garlic root tip cells, we see cells going through the usual phases of mitosis. The orange arrow points to a cell undergoing anaphase, for example. However, when you block the formation of microtubules, they can no longer play their critical role in aligning chromosomes at the metaphase plate and separating sister chromatids during anaphase. Metaphase and anaphase are thus disrupted. In our experiments in garlic root tip cells, we saw this lead to cells stuck in prometaphase and metaphase. You get these cells where the chromosomes are not properly aligned and aren't able to separate.
So, what is colchicine used for today? Well, it's primarily prescribed to treat inflammatory diseases like gout. Gout is caused by a buildup of urate, which is formed when the body has excess purines. These crystals interact with macrophages from tissue that produce lubricants for joints. This leads to an immune response cascade that secretes pro-inflammatory cytokines and neutrophils into joint tissue, leading to inflammation. The primary mechanism by which colchicine is known to treat acute gout attacks, which is not directly related to mitosis, is by impairing the formation of microtubules such that inflammatory cells cannot migrate via chemotaxis. However, colchicine is also known to inhibit mitosis and thus prevent the formation of fibroblast cells. Fibroblasts are a primary source of inflammatory cells. This is perhaps a secondary mechanism by which colchicine treats gout. The standard dose for this usage is 1.8 milligrams daily. Novel uses of this ancient drug are being explored too. In 2023, the FDA approved Ledoco, which is 0.5 milligrams of colchicine, to reduce the risk of cardiac events like heart attack and stroke, and it operates by impairing inflammatory pathways as well. Colchicine is also being studied for use as an anti-cancer agent. However, its use in oncology has been limited by its toxicity at high doses. Through its antimitotic mechanisms, colchicine can trigger apoptosis of cells. Several analogs of colchicine are being synthesized to be less toxic than natural colchicine and are being explored as an option within drug formulations that target solid cancers.
You might be wondering, what else is colchicine used for? Well, it's used to induce polyploidy in plants. Polyploidy is when an organism has more than the normal 2 sets of chromosomes. As we discussed earlier, colchicine disrupts microtubule polymerization, preventing chromosome segregation and leaving them aligned at the metaphase plate. If the cell proceeds to cytokinesis without completing anaphase, it may end up with 2 full sets of chromosomes, resulting in a tetraploid cell instead of 2 diploid daughter cells. A cross between a diploid and a tetraploid can also create a triploid. And these types of cells can be sterile and also can lead to seedless plants. Polyploidy is used to create new plants or new characteristics. So plants might be bigger as a result of more DNA. They could also have thicker leaves and flowers, more intense colors, longer-lasting flowers, and colchicine treatment and other types of plants can improve root and shoot growth and plant biomass. Despite how valuable this can be, colchicine treatment doesn't always induce polyploidy, and it can also lead to non-viable plants. Different dosing concentrations are needed depending on the plant that's used. Colchicine is normally used in an aqueous solution with a concentration between 0.1 and 0.8%. Techniques used include flow cytometry, morphological measurement, and root tip squash, which is what we did in our lab, not to induce polyploidy, but to visualize mitosis under a microscope. It's important that colchicine be used carefully in humans and with plants. Colchicine can cause side effects like anorexia, nausea, vomiting, abdominal pain, and diarrhea. Higher doses have also been associated with colchicine overdose or poisoning, which is the result of the arrest of cellular mitosis. Dosages as low as 0.5 milligrams per kilogram have been fatal. Colchicine is not particularly dangerous to use in the lab with known concentrations, but following safety protocols and using PPE is important. Speaking to medical professionals about side effects is also crucial for those using it. Colchicine is very valuable as long as you use it safely. Thanks for listening.